kill pretty much anything that's alive, including bacteria. So people figured out pretty quickly that if we put mercury onto these open wounds for our syphilis, that it'll kill the syphilis. But then it also means that this mercury that the hatter is breathing in and driving him crazy, we now have thousands or millions of people rubbing mercury directly into their bloodstream. So very often, if you can imagine, instead of breathing in a poison, you're now putting it directly into your body. This was often even more lethal than just living with the syphilis. And so, again, this period of medicine, we're thinking of doctors seriously had to question of which one is worse, just letting you die of the disease or trying to treat it. And that was a very serious question that doctors had to ask a lot 100 years ago. Here comes another fun picture. So this is an infection of staph infection. You might have heard of a staph infection. This is caused by a bacterium called Staphylococcus Aureus, which normally lives on our skin. But staph infections typically, they can cause open wounds, open sores. If you get boils or pimples, that's often a staph infection. Now the treatment for a staph infection 100 years ago, before we had antibiotics, was this. This is a spray for carbolic acid. Now, this is exactly as it sounds. It is acid. We're going to burn the infection out of you. And this did work, but it was not very pleasant. We do know that surgeons about 100 years ago also figured out that this worked for hand washing. So they would turn carbolic acid into a soap, and you could always tell who was the surgeon in a room because their hands would be really raw and red because they were literally washing their hands in acid. This is not a pleasant experience to be getting rid of something as simple as a staph infection by burning it out of your skin. So that's it for all the super gross photos, so you don't need to worry. So the other disease that I'm going to be talking a little bit about is tuberculosis. And so here we have a depiction of tuberculosis in a painting that was done by the Venezuelan artist Cristobal Rojas Pelayo. And tuberculosis remains one of the biggest killers amongst all infectious diseases in the world. But tuberculosis would essentially get into your lungs, and once it would live there, over time, your body was essentially consume itself, which is also why tuberculosis at the time was called consumption. It would be like you would cough a lot, but you would also just become more and more tired, and you would become more and more frail until at one point you just wouldn't wake up anymore. And this was a very, very common affliction, tuberculosis. And the only real treatment for it that we had were these. So on the right, we have a pneumothorax apparatus, which when you go to the exhibition, we actually have one of these, which is really neat. And so a pneumothorax apparatus, if you can imagine, this is like, think of a bicycle pump that's attached to a needle. And you stab yourself, or a doctor stabs you with the needle, and then inflates the bicycle pump like crazy, so that it makes a giant bubble inside of your abdomen, so that you kind of inflate. And as you inflate, it crushes your lung, so that the infection inside your lung would die along with your lung, so that you would then live on only one lung. So not terribly pleasant, again. And then on the left, we have an x-ray of a person that went through a procedure called plomage. Now, this was another very common treatment for tuberculosis that goes by the same principle. Only this time, instead of inflating your abdomen to crush your lung, you can see there are like these little balls here on the right-hand side of the lung. You can see that? So these are plastic or rubber or glass balls that would be put into your lung through surgery. And they would sit there, and the idea is that they would, because they're balls, they rest inside your lung, and that they would deprive the lung of oxygen, and it would kill your lung. And this way, if your lung is dead, so is the tuberculosis. And then you would just live the rest of your life on one lung. So we're just looking at a very dark part of history in terms of medicine. Medicine was not great. And again, this is only about 100 years ago. If you can imagine, this is after World War I. So this is after industrialization. This is just how probably your great-grandparents would have known medicine. So these are only just a few examples of these kinds of medical treatments and the kinds of diseases that were very common. But it doesn't even get into other diseases that really struck people down by the millions. Things like cholera or tetanus or gangrene. And if I want to just say one thing, is that before we had antibiotics, medicine was horrific, cruel, and very, very bloody. And it's all because of these guys. Oh no, this is another thing. I'm not ill prepared. I just surprised myself with my own work. So this is bloodletting. So this was another potential treatment for tuberculosis. So bloodletting was used right up until the 1930s, which is very alarming. And it was used for all different kinds of ailments, even for things like migraines. But the thinking was that if you had an infection in your blood, that if we got rid of a lot of your blood, then maybe the infection would go away too. And again, this was used right up until the 1930s. So this isn't something that's from a whole other time. This is actually quite recent. We do know that bloodletting theoretically worked in that it made the patient feel better, because you would go to your barber, not too bad, you would go to your barber in order to get this done. Get your hair cut and then your arm cut, normal. They already have the knives. So so you go to your barber, they put a little nick in your arm, and then as they take out blood, the patient really does start to feel better. And now we think they start to feel better because they would be losing so much blood that you sort of feel a bit woozy after. It's like, oh, I don't hurt anymore. How great is this? So we do know that patients did feel better for a day or two, and then once all their blood was able to replenish and the infection came back, they would just feel bad again, and then they'd head back to the barber. So this was a very, very common practice. And actually, has anybody heard of George Washington before? Yeah, first president of the United States. He got a blood infection while he was in office. And he died because he had an infection, and his doctor thought, oh, really, the problem is you're not doing enough bloodletting. We just got to do more bloodletting, more bloodletting, more bloodletting. And so we had 6 sessions of bloodletting over the course of 3 days. And so he didn't die from his infection. He died because he was a stuck pig, as they say in the UK. Like, he was just drained out of all his blood, and he died from blood loss. So this was a very common treatment for tuberculosis that now we know definitely didn't work. And what makes it even more awkward is sometimes they would even make infections worse, because you are opening up a wound in your body, and so it makes it very easy for bacteria then to get into more bacteria to get into your bloodstream. So we knew that people would often pick up infections while they were at their barber, and then they would die from a treatment. So just again, you have your treatment, you have your disease, and it's kind of a toss-up as to what's going to kill you if we go back in time. Now, as I was saying, this is all because of these guys. Yeah, there we go. That's the right slide. These are bacteria. Has everybody heard of bacteria before? Yes? Fantastic. These are my people. So bacteria are single-celled organisms. They are their own kind of animal. And they have their own lives as well. They live everywhere that we have looked for them. They're very good at adapting to their environment, so we've found them. They've been around for 3.5 billion years, I should say. So they've been evolving for that entire time. They're some of the first creatures to actually evolve to be alive. And in that time, they've become evolution machines. So we found them on the very edge of Earth's atmosphere, on the border between the atmosphere and space. We found them miles underground, getting crushed by Earth's pressure. We found them at the bottom of the ocean. We found them in volcanic vents. Everywhere that we look for them, we find them. Even in places that we think there's no way that anything living could be here, we have often found bacteria. So these bacteria are also on and inside of us, which is really quite important. They've been here for a very, very long time, and they'll be here long after we're gone. And as single-celled organisms, they are their own kind of animals. They live their own lives. They hunt for food. Sometimes they forage. They make families, they build communities, they make friends. They have a really weird way of talking to one another. They also have a really weird version of sex, which I'll get into later. Talk to your parents about that. So they do have their entire own lives, and they even have their own ecosystems, just like we do. So when we think of bacteria, we shouldn't think of them as just being like these little things that are dirty, that are just here to kill us. We should be thinking of them like you would think of like a bird. A bird, is there a good or a bad bird? Well, not really. It's either a bird or it's not. It's just kind of there. And we should be thinking about bacteria in the same way, except this is a giant bird that also has the capacity to kill us. But that's very cool, is what I'm trying to say, as opposed to something terribly scary. So again, bacteria have been around for 3.5 billion years, and they're very, very good at changing themselves to suit any kind of environment they're in. They also are very good at changing their environments to suit themselves, much like we are. we're not very good at living in jungles, so we build houses. Much in the same way bacteria are like that. The reason that we have oxygen, we first got oxygen into the atmosphere on Earth because of cyanobacteria, which is a type of bacteria. These bacteria actually produced the first injection of oxygen into the atmosphere so that they could move beyond just the ocean. So we are very much living in their world. They're not living in our world. They are the dominant species. If we had an alien that came to Earth and was doing his report back home, they would say, well, you know, there are these humans that are there, and they think that they're in charge, but really there are these millions of species of these tiny things that are absolutely everywhere and have completely dominated the planet for billions of years. So these guys, these are the ones that are really in charge. But because they've been around for so long, we're still the new kids on the block in terms of evolution. But that means that we've also evolved alongside of them. So this is my very favorite bacterium. Everyone has their favorite, of course. I won't ask you yours. I know you've got so many to choose from. But here we have Masseria Gonorrhea, which is my personal favorite. It often afflicts people as the disease called gonorrhea. But I really, really like Masseria Gonorrhea, not just because they look like little bums, a few million years ago, these bacteria, they originally come from swamplands. They used to live in mud, and at one point they made the move from mud onto primates, or pre-humans. And once they made that move onto humans, they never left. To the point where now we've evolved together so much that the only place in nature that you can find Masseria Gonorrhea is on humans. They can't survive anywhere else. They have been our partners in evolution ever since. So the only place on the planet to find Masseria Gonorrhea is on human genitals, which is just a whole lot of fun. Also to the right there, you can see something really cool. This is part of what makes Masseria Gonorrhea one of my favorites. So if you can imagine, like, you guys can imagine what a sperm cell looks like, how it's like a cell that it has like a little tail. Yeah, that's the universal symbol for sperm. If you can imagine, that little tail is called a flagellum. And bacteria have these flagella as well. It allows them to move through different kinds of surfaces. But when bacteria develop flagella, Masseria Gonorrhea develop flagella because they live on surfaces instead of in liquid. So when they live on a surface, they have to grow out these flagella to drag themselves across their skin in order to get to food sources. And these flagella pull themselves on biological motors, not like muscles like we have. And these motors are so strong that they can pull 200,000 times their own body weight, which makes them the strongest thing on Earth. This is the equivalent to one of us being able to pick up a Boeing 747 plane. These things are very, very cool. And now, I hope gonorrhea is your favorite too. Of course, and not just because they're little bones. But I think this also demonstrates that bacteria are very much a part of us and they're a part of our evolution. And they're also a part of our biology. So as I mentioned before, they are our partners in evolution. In that these are not found anywhere except for humans. In the same way, you cannot find a human without bacteria. So we know that when you are first born, the only time that you are sterile, when you have no bacteria, is when you're still in your mother's womb. As soon as you are born, as you're being pushed out of the birth canal, you actually pick up a colony of E. coli bacteria from your mother in the birth canal, which is really fun. It's like the last gift that she gives you as you're being born. Well, okay, birthdays or whatever. But, like the most important gift that she gives you is E. coli. And in theory, that E. coli lives with you for the rest of your life. Now, as soon as that E. coli enters your gut, which is where it lives forever, you should now all have E. coli in your gut. Thank your moms. Once it enters your gut, it sends a signal to the rest of your body saying, you are now in the world, it's time for you to turn on your immune system. And then it starts kick-starting all of your biological functions. So this infusion of bacteria is really, really important for us to make the change from being a fetus into being a baby. Or being born into being a human, or however you want to make that distinction. So this is a part of what makes us us. We also know that all throughout our lives, these bacteria are able to send us signals. So when you get certain kinds of cravings, we actually think that we don't have one brain. We actually have two brains, one of them in our noggin and the other one in our gut. And the one in our gut is controlled by bacteria. So they can actually communicate with us so that when we get cravings for certain things, a lot of that is actually our bacteria sending us signals. So if we find ourselves to be particularly like, oh, I really want something salty, or I really want something sour or something, that's your bacteria speaking to you, saying, this is what we want in your gut. Feed me. We do also know that we've evolved to support these bacteria. They're meant to be inside us. So again, right after we're born, we have these E. coli living in our gut. In your mother's natural milk, the milk that she produces has sugars and proteins that cannot be broken down by the human digestive system. They can only be broken down by bacteria. And that tells us that not only have we evolved to need these bacteria, but these bacteria have evolved like a symbiotic relationship with them. We're meant to have them. I keep doing this because the bacteria are like in our guts. Yeah? Okay. I'm just making that clear that I just have a weird thing with my belly button. So, yeah, so these bacteria are really, really important, to the point where they are so inside of us that about 53% of all the cells in our body are actually bacteria. They're not human cells. So we shouldn't be thinking of ourselves as just being humans. We should be thinking of ourselves as these microbial jungles that happen to be walking around and take the form of a human. That's really how we should be thinking of ourselves. We should be treating these guys with respect. They are very much, very, very much a part of us. In fact, it was up until about 2 years ago, we always think of things like urine as being sterile, as being like, oh, there's no bacteria in urine. But just 2 years ago, there was a study done about how apparently there are about 54 species of bacteria that just live in our bladders. So we know that even when we think that things are sterile, they're not. They're full of these bacteria. They're a part of our life, and it's just a part of the human experience. But to take things one further, not only do they impact our biology, but it also impacts our culture and our history, and even our economics. Oh, you've got a question. That's right, you can make things sterile artificially, but naturally speaking, you won't find a surface that's just, oh, this is a sterile surface. That's a great point. You can artificially make things sterile, but it's an artificial thing that we're doing on purpose, as opposed to something that naturally occurs.
Salim, just a moment. If you have questions, please hold your breath. After the lecture, we're going to have a session exclusively for your questions, okay? Please don't interrupt him. I'm sorry. Thank you.
We do know that the history of bacteria are very much interwoven with the history of humans and with our culture. So if anybody's ever had yogurt or cheese, you have gone out of your way to consume a food that was made by bacteria. If you ever eaten pickles, for example, these are made with bacteria. So it really impacts our diet. It also impacts our history. You may not have heard of the bacterium called Yersinia pestis, but this was the Black Plague, which killed off a third of Europe and completely changed how economics and government would work, which eventually paved the way for this little thing called Western-style democracy, which is always good. And then it even changes how we practice our religion. So this is another really fun bacterium. This is called Serratia marcescens. So this is a bacterium that, as it clumps together, it produces like a slime, like a really reddish slime, and it typically grows in bread and in really wet surfaces. So here's Serratia marcescens in bread. So if we go back in time to the year 1263, to a little town in Italy, there was a church that was getting ready to do communion, which is a Christian ritual where a priest offers bread to everyone in the congregation as a way of saying, this is a piece of Jesus Christ who has died. It is a part of his body, and then everybody eats it. But as they were handing out this bread had an infection of Serratia marcescens. So as they're handing out this bread saying, this is the body of Jesus Christ, they see the bread start to bleed because it's full of this bacterium and it's dripping with red. And they see it as a miracle. And it's declared by the church as an official miracle, and even today that church is now it's a pilgrimage site for people to visit. And so it's made its way into our religious function even, this religious observance of bacteria, which I think is really neat. So As I mentioned before, bacteria are very, very good at evolving to any kind of environment they're in. Anywhere that we put them, they find a way to survive over time. But they also find ways to fight one another. So I mentioned before that they have a very complex lifestyle. They hunt one another. And they do fight one another for resources. And that all plays into the birth of the age of antibiotics. So if we go back in time to 1928, I guess we're going forward, because we were just in 1263. Wow! And so if you move forward to 1928, we go into a little biology lab in St. Mary's Hospital in London. And there was a researcher there named Alexander Fleming. And at one point in September, he was doing a series of experiments. He was very messy, and he didn't tidy up after his experiments. And he left for the weekend to go on holiday. And when he came back, he found the mess that he had made still. Wouldn't you know it? Messes that you make don't clean themselves up. But when he came back from holiday, he found that there was a mold growing on a petri dish that he was using to grow bacteria. And not only did he see this mold, but this mold was also killing bacteria that he was trying to grow in that dish. This hadn't really been observed before. And so he wrote about it in 1929. There was a big hullabaloo about it. And he named that chemical that this mold was producing to kill bacteria, he named it penicillin. Now on the left, this is what penicillium, which is what this mold is. The mold is named Penicillium notatum. This is what that mold actually looks like. You probably have seen this mold before. It's very, very common. It often grows on old fruit and bread. So if you leave old fruit out in your kitchen for too long, it'll eventually turn like a green with white streaks. That is penicillium, which has saved hundreds of millions of people's lives, which is really neat. But this mold, just like we do, has its own immune system that it uses to fight infection. And it was that immune system that Alexander Fleming observed, saw that it's a chemical, and named it penicillin. Now on the right, you can see what penicillium looks like down a microscope. The Latin name of penicillium means a paintbrush. So you can kind of see where that comes from by looking at it down a microscope. So he named this thing of penicillin. But penicillin had been used for thousands upon thousands of years. So we do know that the ancient Greeks, the ancient Indians, the ancient Chinese, and someone else did it. Did I say ancient Egyptians? Maybe, maybe not. But ancient civilizations, when they rode into battle, they would take with them moldy pieces of bread. And they didn't understand fully why, but they knew that if you got shot with an arrow or stabbed with a sword, and this is advice that you can take home with you. If you get shot with an arrow or stabbed with a sword, that if you dab yourself with moldy bread instead of just with a regular bandage, that those people would tend to survive better. Now they didn't understand it at the time, but now we know that they were using penicillin, which is really very cool. So penicillin is something that we've been using for thousands upon thousands of years, and now you know how to treat an arrow wound the next time you get caught out. You're welcome for that piece of advice. That one comes for free. But immediately afterwards, Fleming abandoned his research into penicillin. He thought, this is an interesting little academic side note. But he thought, this isn't going to go anywhere. This is a whole lot of work. And he completely changed his research and decided to go into the study of lysozymes. Now, lysozymes are chemicals that are in a lot of different living things that naturally kill bacteria. We have lysozymes in our tears. Which is really fun. If you collect enough of your tears, it means you can actually kill your own infections, which is pretty cool. It's also why if you come to London, I highly recommend it. It's a lovely place when it's not raining and cold. But if you come to London in the bunkers where Winston Churchill was hiding during the Second World War, commanding, yes, where he was commanding. In those bunkers, you can find a vial of Winston Churchill's tears, because he was actually allergic to penicillin, so he couldn't use penicillin, but he wasn't allergic to his own lysozymes, so that if he got cut or injured or something in the bunker, he could douse himself in his own tears, which sounds weird, but it also means that at the time, there were a lot of great political cartoons making fun of Alexander Fleming, because when Alexander Fleming found this, overnight he became world famous. But then once he abandoned that research, it became a laughing stock. Once he switched to lysozymes, we got really great cartoons like this one, where you can see Alexander Fleming whipping children in his basement to collect their tears, which I just think is really fun. And it also means, if some biology textbooks, you get some instructions on how to collect tears from different animals, which I think is a also really fun. These are just kind of illustrations you just don't find anywhere else. So this takes us to the year 1942, and we have an Australian visiting professor in Oxford named Howard Florey, and a German-Jewish refugee professor named Ernst Chain. And they're having a conversation, I imagine over tea, but most likely it just consisted of one of them walking to the other one's office, saying, I read this paper all the way back from 1929 that said that there's this thing that can kill bacteria. We're in the middle of World War II. We must be able to prevent some of these infections that are happening in all of our soldiers. We must be able to help the war effort with our medical science backgrounds. So they decided, yeah, sure, let's give it a go. So they teamed up. So this is a picture of them in 1945. But the guy with the fantastic mustache, that's Ernst Chain. I think he looks a little bit like Einstein in the early years. And then on the right, that's Howard Florey, the Australian professor. They enlisted the help of these two guys. The guy in the middle is Norman Heatley, and by guy I also mean woman on the right. But Norman Heatley his focus was on how fungi grow, and the woman on the right is Dorothy Crowfoot Hodgkin, who is the unsung hero of penicillin. She does X-ray crystallography, which means she uses X-rays and mathematics in order to figure out the structures of molecules, so that as they were going through these experiments with penicillin in order to turn it into a human medicine, that maybe she'd be able to figure out the structure of it in order to make more antibiotics. Weird. So she's amazing, but of course got snubbed for the Nobel Prize. Nobel Prize Committee! But again, I cannot stress this enough that Alexander Fleming disappeared into obscurity. He was researching his tears and whipping children in his basement. He was a nobody at this time. And in fact, Howard Florey turned to Chain and said, wait, we're doing research into penicillin based on this paper. Don't you think we should involve the author of this paper, the person that discovered penicillin? And Ernst Chain's response was, wait, you mean Alexander Fleming? Isn't he dead? He had been out of academia for so long that he had just disappeared into obscurity to the point where they thought that he was dead. Yeah, make sure you keep publishing if you go into academia, folks, because... So it took about 2 years of work in order to turn it into a human medicine. And in 1945, these three guys won the Nobel Prize for it. So on the left is Alexander Fleming, and then we have Ernst Chain and Howard Florey. Yay! But Norman Heatley and Dorothy Crowfoot Hodgkin were completely snubbed for the Nobel Prize, which is really quite tragic. Their names really should be going down in history too, which is why I'm mentioning them now. They're heroes and wonderful and saved hundreds of millions of people's lives. So right after we got penicillin, it was a complete game changer. This completely opened the door for new kinds of medicine. Because now that we're able to fight basic kinds of infection, we can do things like surgeries, which we couldn't do before. So we could do organ transplants, we could do hip and knee replacements, we could do chemotherapy, which is one of these things that we just kind of take for granted, I guess. We could implant pacemakers in case you get a heart attack. We get protection for premature babies, because about 5% of us are born premature. It directly saved the lives of 200 million people, but indirectly saved hundreds of millions more. And right after penicillin came out, people went absolutely nuts for it. When you find a miracle, why would you want to hold it back? So penicillin made its way into lipsticks and cosmetics and food and all kinds of things that you'd find around the house. So on the left is the lipstick that was an Estee Lauder lipstick that is infused with penicillin from the 1950s, which is super weird. And then as penicillin really took off, almost overnight, it added 20 years life expectancy onto our lives. So if you can imagine the kind of impact that would have, if you're going through life thinking there is no way that you're going to live past the age of 55, and then you're told, well, no, you're good at least till 75. That'll dramatically change the way you live your life, you think about the future, the way you have your family. This changed the world, not just the world of medicine, but just everyday lives of people. The idea that suddenly, if you're born premature, you're not just going to die, was a really, really big deal to a lot of families. Hopefully, for all families, probably. The success of penicillin as well led Dr. Anthony Fauci, who was a world leader of pathogens in the 1960s and 70s, to say, even with my great loyalty to infectious disease, I cannot conceive of the need for students in infectious disease unless they intend to spend their time culturing one another. So there was very much this culture of like, we have infectious disease in the bag, we've got it figured out, we can just sort of move on, we don't need to worry about all these diseases that I talked about at the beginning. But then we have to come back to this guy. Remember this guy, these are bacteria, so these specifically are E. coli. If I can give a sense of scale, by the way, if you can imagine the sausages of bacteria. From one end of the sausage to the other end of the sausage is 2 microns. 2 microns. The width of a human hair is 20 microns. So we can fit 20 of these bacteria across a single human hair, which is these are very small. And these are also the giants of the microbial world. Most bacteria are significantly smaller than E. coli. So, we're returning back to the bacteria. And remember how it's very good at evolving to its environments. And now that we're starting to fill its environment with penicillin, and about 30 other antibiotics that we've discovered since penicillin, they're able to evolve resistance to their new environment, which is full of penicillin, and they become resistant to antibiotics. And so we're generally losing these miracle cures. So these 20 years of life expectancy that was added on is now starting to disappear because these bacteria are so able to change. Now, they're able to do this in 2 ways, and the first is evolutionary pressure. I'm going to show you what that looks like. So you can imagine, here we have a colony of bacteria. Aren't they beautiful? Just wonderful things. We love these bacteria. They're great. They're inside of us, all around us, and all of that. And just naturally, these bacteria will be genetically different from one another, even if they're the same species. It's like you and I are the same species, but just naturally, we're going to develop certain differences. We're going to have different colored eyes, for example, just because that's just the way that DNA rolls. And so naturally, some of these bacteria might mutate or just evolve so that they have some kind of a differentiator that makes them resistant to antibiotics. So antibiotics like penicillin interact with bacteria by messing up with their biology in different ways. So maybe this kind of a mutation is something like, their skin is a little bit thicker, so it's harder for the antibiotics to get inside the bodies. Or maybe this mutation is, our waste product, the thing that I exhale, the thing that I breathe out, is actually dangerous to antibiotics. It destroys antibiotics before it can reach them. So they just sort of naturally have a general mutation. But then, if this is in our body, we don't like an infection that we have, so we then use antibiotics, and it kills everything off, except for the resistant ones. So the resistant ones are the only ones left, because they naturally have this resistance, just kind of randomly, as a part of a mutation. But now you have a colony where 100% of the bacteria, even though there are only a couple of them, are resistant. And over time, they multiply and multiply and multiply until you have an entire colony that is now grown, and bacteria can multiply every 20 minutes. They can divide. So over not much time, they can become a very big infection that is no longer affected by antibiotics. And what's really weird is that as they develop these kinds of mutations and these kinds of resistances, they can share them too. So one of the cool things about bacteria, well, we think of our cells, because we're made up of many different cells. We have trillions of cells in our bodies. So it's very difficult for us to change the biology of our bodies. Our genetics are pretty much set. But bacteria are only made of one cell. So if they need to make a change to their DNA, they only need to change one section of DNA in order to make it throughout their entire body, because their entire body is only one cell. So they become very good at changing their DNA because it's just so easy for them, and they can do it in a lot of different ways. Now, our DNA is kept inside of the nucleus of the cell, and it's in very long strings. But in bacteria, it's kept in little loops. And what's convenient about these little loops called plasmids is that they can multiply them and spread them out into their own environment. So here we have bacterium on the right. Or maybe on the left. So first, the bacterium on the bottom, the saucy one, it's linking up with 2 other bacteria because it says, I have DNA that I really like, that I think you will like too. So it extends like a weird little bridge to these other bacteria. It says, here, have this DNA that I have found to be very helpful. Now this DNA might be anything. It might be, now I'm able to eat something that I couldn't eat before, so I can get energy easier. Maybe it's something like I can survive in hotter temperatures that I couldn't before. But what's really dangerous is that they can also show these genes that say, well, now I can survive antibiotics, which I couldn't before. So here it's trading DNA with its buddies. And then on the right, you can see this bacteria that's going fishing. So the green is the bacterium, and it sees that there are lumps of DNA in red all around it. And it says, ooh, I could really go for some extra DNA. And so it starts doing a little bit of fishing, a little bit of shopping. It says, yeah, a bit of that, a bit of that, a bit of that. And then it collects those, and then puts it into its own body so that it can change. And they can do this across different species, which is what makes it really cool. So if you can imagine, you're walking down the street, and you see a pigeon, and you say, oh, I'd like to look at that pigeon. And you grab the pigeon and you rub the pigeon against yourself, and then you sprout wings. We think of that as being completely ridiculous, but for bacteria, they are doing that all the time because they can do this across species and they can fundamentally change their own DNA, which is something that we really couldn't even imagine doing to ourselves up until just a couple of years ago. And so it's very, very cool. But since we're using so much of penicillin in the 50s, 60s, 70s, 80s, 90s, we're using penicillin, we're using all different other kinds of antibiotics, and we are filling their environment with antibiotics. So not only are they evolving naturally and sharing these kinds of resistance with each other, but they're also going through this process of evolutionary pressure, which I spoke about before. We're just killing off everything that isn't resistant, so the only thing left are the stuff that is resistant. And so these old diseases, which we thought had been relegated to the history books, are now making a comeback. So we have diseases like tuberculosis, which was completely treatable since 1953. It's now making a comeback with a vengeance because now it's resistant to antibiotics. Gonorrhea, which was normally cured with just a 3-day course of penicillin, is now untreatable in a lot of areas with any antibiotic, causing widespread infertility and complications with kidneys. And the bacteria are really making a comeback now. And this is particularly a problem here in India. I think this might be a good time, just because I am now moving on from bacterial biology. If there are any questions specifically about bacteria and how bacteria work, just to go through very, very quickly. No?
Yeah.
Everyone understands 100%? I love it! That's the best. I'm either doing a great or terrible job. Let's assume great. I like how we're all on the same page. When you go home to say, oh yeah, Sheldon, he was an amazing speaker. Wasn't he so great? I don't know. So this problem of antibiotic resistance, this is especially an issue here in India. And it's, oh, sorry.
Sorry to interrupt. You asked for review of this, right?
Sorry?
A go-through of the bacteria, what he explained. Can you please again repeat it?
Oh, yeah.
Can you please repeat the bacterial story again? How it is
From the top, here we go.
That's what you asked.
Let's start at the very beginning.
No, after...
I'm glad you thought it was so engaging.
No, not from here, the last part. This is the
Just
last part.
this bit.
The TBI, how you connected it with TBI, how it's coming back.
Okay. So,
That part.
so it's because of this evolutionary pressure.
Yeah.
So if you can imagine, you have a case of TB. Not making any accusations here, but let's pretend you have a case of tuberculosis. And naturally, some of the cells in your case of tuberculosis are going to be resistant, because that's just how they work. They just mutate every now and then, that's just how they work. But you have tuberculosis, so you go to your doctor, and your doctor prescribes you antibiotics. And the antibiotics kill all those bacteria, except for a couple of the TB bacteria that are naturally resistant. Now we've killed off all the other bacteria. The only thing left is the resistant one. And now you have a resistant case of tuberculosis. And now that you have a tuberculosis that's fully resistant, if you pass on tuberculosis to anyone else, it'll be the same kind. It'll also be resistant. So if you have an infection that passes on, it is now untreatable, which is not cool. And what makes it worse is that if you take, let's say you go to the doctor and you don't need antibiotics in the first place. So you do this process, but with the E. coli in your gut. I'm sorry, Mom. So you kill off all those E. coli that your mom gave you as a gift. How ungrateful. You kill off all the bacteria in the body. that only the E. coli I left. So sorry, Mom. Don't tell her. But then they grow, but it's fine because you're meant to have E. coli in your gut. So if the E. coli becomes resistant, that's not a big deal. But let's not forget that bacteria also do this. So if you have resistant bacteria as E. coli, which is fine, because why would you want to kill off your E. coli? It's from your mom. You're not trying to kill off your E. coli. But you do want to kill off your TB. It's too late, because the E. coli and the TB have already been scheming, and they've been swapping their DNA with each other. And the E. coli has been creating all this DNA and spewing it out. So it doesn't even matter that the TB didn't specifically become resistant. It matters that other bacteria in your body did. So even your buddies, even the bacteria that you want to be there, are still scheming against you. And so that's how we land up with these That's how we land up with these diseases. Again, I'm not unprepared, I'm just surprised by my own work. That's why we land up with these diseases like TB making a comeback, because now they have become what Alexander Fleming called educated microbes. That they're these bacteria that have learned, not only from their environment, but from one another, because we've been overusing these antibiotics. Cool. I think I'm going to move on. Is that okay? I'll have a question here at the end, I promise. Hold on. Don't worry. I'll come back. I will burn your face into my brain. All right. So we do know that this problem of antibiotic resistance is especially pertinent here in India for a few different reasons, and I'm going to be going through why. But firstly, the biggest thing that makes India so unique in its consumption of antibiotics is just how much we are consuming. So there are higher rates of resistance in India than anywhere else on Earth. And up to 4% of bacteria in India are resistant to all available antibiotics. 46% of all instances of Staph aureus, which is the bacteria that I showed you before that's on your skin, that we all have on our skin, 46% of Staph is already resistant to methicillin, which is a type of antibiotic. You might have heard of this methicillin-resistant Staphylococcus aureus, or MRSA, in the news. So about 46% of us have superbugs on our skin right now by being in India. And this also extends to the environment. Oh, sorry, I actually skipped ahead. Oh yeah, sorry. We're overusing antibiotics. We're overusing antibiotics in India like nowhere else in the world as well. So in India, we're using about 13.6 doses of antibiotics per 1,000 every single day. 13.6. Bear that in mind. 13.6. You compare that with China, where it's 8.4. And the equivalent of 18.4% of the population in India will take antibiotics this year, 18.4%. And this is better than some countries, like in Turkey, where it's 44%. It's significantly worse. India's at 18.4%, and we're trying to get this number as low as possible. In Northern Europe, it's as low as 3%, and even regionally, like in Pakistan, it's at 9%. So even regionally, other countries are able to bring this number way further down than we're able to in India. So there's a lot of antibiotics being used, making it easier for bacteria to develop resistance. This is spurring on what we had before, about this idea of having selective evolution, of evolutionary pressure. And they're being used so much because of availability. So we're not supposed to be able to get antibiotics just from a pharmacist, but how many of us have gone into a pharmacist or with our parents maybe and just said, oh, I have this thing. Okay, I'm going to use antibiotics. It's very easy to get antibiotics, which is why we have so much antibiotics being used. In India, this is the right slide. This is the right one. In India, we've doubled the amount of antibiotics that were being consumed per person between the year 2000 and 2015. And if we don't do anything now, that number is going to triple again by 2030. So if we know India is using so many antibiotics, do we know that this is actually leading to resistance? And the answer is yes. So if the problem we've identified that makes India unique is that we're using so much antibiotics, there's a lot of antibiotics flowing freely around, it's very easy to get antibiotics, and we're using more and more and more of it every year. So does this actually translate into resistance? The answer is yes. So this is where I'm meant to be talking about this. We have higher rates of a resistance for certain bacteria in India higher than anywhere else on the planet. Up to 4% of bacteria here are resistant to all available antibiotics, and 48% of all staph is actually MRSA, which is very, very high. And this also extends into the environment. So we're even pumping antibiotics into our environment, not just into people. There are about 250 companies here in India with factories that are producing pharmaceutical companies. And right now, they are almost completely unregulated in how they dispose of their waste. So it means that as these companies produce antibiotics, the waste that they make from it just goes straight into river systems. And so we're seeing antibiotics in river systems and in water and in soil, and that's just making resistance even worse in India, that then can translate into diseases in humans becoming more resistant, as we looked at before. And this is really serious. In river systems, there's a 10 times higher concentration of antibiotics in river water waste in India than anywhere else in the developed world. There's a higher concentration of antibiotics in river water in Hyderabad than there is in the bloodstream of a person taking antibiotics. In 2012, there were 283 colonies of E. coli were sampled as a part of a study in Karnataka, and every single sample was resistant to at least 3 different kinds of antibiotics. So we know that there is a crisis of resistance in terms of we know that there are 2 crises happening so far. We know that we are using a whole lot of antibiotics, and we know that we're seeing resistance appearing in our own environment, in our water, in our soil. And this can also have disastrous effects for the whole world. In 2006, there was a new plasmid. So before I talked about these plasmids, these loops of DNA that helped to create conditions for resistance. One of these was specifically found in New Delhi, so it was given the very, very attractive name of New Delhi metallobeta lactamase 1, which is, let's be honest, the best of all the New Delhi metallobeta lactamases. So it was given this name of New Delhi metallobeta lactamase 1. Try saying that 100 times fast and we'll get to the same level. Or NDM1. So this was a brand new plasmid that was identified in 2006, saying this is a gene that causes antibiotic resistance. We found it in India. And after it was first discovered in India, we then started keeping tabs on it to see where else it would pop up. And every country that we've looked for it, we've found it after India. So we know that whatever starts in India happens to go throughout the world. Because the most important thing to remember about antibiotic resistance as a global issue is that bacteria do not care about borders. An issue in China is an issue in Mexico, is an issue in Brazil, is an issue in Ireland. Bacteria do not go through customs. They go through water systems, they go through people, they go through shipping containers, they go through absolutely everything, and they do not care. They blow in the wind. They can very easily spread. So when we think about antibiotic resistance, we should not be thinking of it specifically as if India gets the world, we should be thinking of it as we are a global player as people in India that can have a really big part in how antibiotic resistance affects the rest of the planet. And we do know that this kind of resistance can also spread into people very easily. So in 2014, there was a study of Swiss travelers. Let's see if I can remember how many it is. I can't remember how many it is. But there were a group of a couple hundred travelers from Switzerland that were coming to visit India. And they had their entire microbiome sequenced. They wanted, these researchers wanted to figure out, okay, you're going from Switzerland to India, let's see how much of the bacteria in India you pick up just by being in India for 2 weeks. So they did a full scoping of their microbiome and said, great, we know what you look like before, and then after 2 weeks they come back and they did a full look after, and they found that 87% of the travelers that came back had bacteria that were then that had NDM1. So we know that this travels on people. We know that it gets into people, and then it will potentially affect their health. These 87% of travelers in Switzerland now have this gene in them to potentially make the infections that they get in the future far worse because they're not able to treat it. So, let's break this down a little bit. We know that India is using a lot of antibiotics. We know that this is leading to resistance because we can track it through the environment. And we know that it can easily find its way into people, and not just people here, but people all around the world. So how do we actually address this issue? We're going to turn from death and destruction to glimmers of hope. How do we save the world? 3 easy steps. These are really good life lessons as well, just if you want to pick these up. Step number 1, prevention is the best cure. The best way to not get an infection, or to cure an infection, is to not pick it up in the first place. It just makes sense, doesn't it? If we can reduce how much antibiotics that we actually need, we'll use less and resistance will go down. And we've actually seen that happen in other countries, like in the Netherlands and the United States. Prevention does actually work. Now, if we think of what's the best way that we can have prevention of using antibiotics, the biggest hotspot of antibiotics in India is in hospitals, and around the world, really. Everyone in hospitals is already sick, so a lot of them are already using antibiotics, and a lot of them have infections that they really don't want to have in the first place. So you naturally have a lot of bacteria, a lot of antibiotics floating around in hospitals. But it also means that it's very easy for bacteria and antibiotic resistance to pass from one person to another. Patients. And this is especially a problem here in India because the hygiene practices within hospitals aren't quite up to scratch. So we know, for example, that 70% of hospitals in India reported reusing surgical gloves in between procedures. We know that 85% of hospitals did not wipe down surfaces after someone gave birth in the room. We know only 32% of doctors washed their hands after handling patients, and 20% did not wash their hands after performing surgery. These are very, very basic interventions. Soap is not expensive. It doesn't take long to wash your hands. It should take 30 seconds. You sing happy birthday to yourself twice. It takes 30 seconds just to wash your hands with regular soap. You don't need a special soap. But this is a very standard intervention that is not being followed in hospitals here. We know that according to the World Health Organization, if you go into a hospital, as a global standard, we expect, just because you're going to be surrounded by sick people, you have about a 10% chance of picking up an X-ray infection, because that's just kind of how it works. You're surrounded by sick people. But in India, depending on which hospital you go to, that odds of picking up a secondary infection isn't 10%. It's anywhere from 11% to 87%. So we know that these infections are passed around amongst patients within hospitals. And that's really, really dangerous, not just for the patients, because it also means that for every extra infection you pick up, you have to take more antibiotics and more antibiotics and more antibiotics. And eventually there will be a point where we don't have access to those antibiotics anymore, and all those people will be really, really stuck in how to provide their own health care. Another big problem here is that there aren't enough doctors and nurses. So according to the World Health Organization, we should have about 25 doctors and nurses per 10,000 people, but in India it's only at about 12 per 10,000. And that means that naturally the standards have to be relaxed in order to make sure that they can get numbers up in order to be able to treat everyone. And that's a really big issue as well. So this is step number 1, prevention. Let's try not to use so much antibiotics as a plan. Step number 2, we can't fix what we don't understand. So despite so much antibiotics being used in India, we don't really know where it all goes, and we're missing a lot of that data, a lot of that information, so that we can't plan for it. So as one example, we know that colistin is regularly used as growth promoter. This is an antibiotic of last resort in humans. It's regularly used in animals. But we don't know how much it's being used. If we just got an understanding of the quantities and of geography that are involved in this, we might be able to plan for it and be able to plan for the future. But we don't have that data, so we can't. It just means that there's no way that we can address a problem that we can't quantify and see into the future. We don't know how urgent this is. We also know that 27% of tuberculosis cases happen here in India, but we don't know how many of those cases are actually resistant. Depending on what paper you look at, it's either 0.3% of tuberculosis patients, or it's, what's that number? 60%. Now that's a very, very big gap. If you did a test at school, and your teacher told you, don't worry about your test, I just graded it, but I have graded it and it's done. And you say, well, what did I get? You're like, and the teacher says, I can't quite remember, but I know it's somewhere between 0.3% correct and 60% correct. Now you're like, well, which one is it? You're freaking out, right? Like, that's a really, really big difference. And then the teacher would say, well, I don't know, it's between those. That's not enough There's not enough accuracy. So we don't know the scale of this problem in India. We don't have the data. So without the data, we can't plan for it. There's also a big issue. If you remember looking back at the can't go backwards. That's okay. We never go back. If you think back to NDM1, the best of all the NDMs, the New Delhi Metallobatal activity. When this was first discovered and it was first published and talked about, the response from the Indian government was, I really, really don't like that you discovered this here. Could we not just change the name? I don't want you to talk about it as long as it has this name of New Delhi attached to it, because then it makes it sound super Indian, and we don't want this associated with India. And it's tradition in academia to name a plasmid after the location we found it anyway. But that's the wrong conversation to have. If you go to your doctor, and or if I go to my doctor, and my doctor tells me, you have this new disease that we just discovered, I haven't seen it anywhere before, so we're going to name it the Sheldon disease. It makes your eyes melt. My first question isn't going to be, could we not call it the Sheldon disease though? My first question is going to be, how do we slow it down? How do we not get melting eyes? How long do I have with my eyes? But instead, the response from the government was to shut down the conversation and say, let's not talk about it. I don't want you talking about it as long as it has this name. And that was the wrong approach to take. And it's a little bit political for me to say, but it was the wrong approach to take because it meant that we lost really valuable years in being able to predict how NDM1 would spread, and it potentially cost a lot of lives. This is, yeah. Oh, I can't go forward. Could someone press a clicky? Yeah, forward. Always forward. Cool. All right, step number 3. Let's set goals for the future and stick to them. So the government signed the Chennai Declaration in 2012, which was a really great step. And this Chennai Declaration said, it laid out like different steps about like, we're going to commit to doing this, and this, and this, and this to make sure that antibiotic resistance doesn't spread. And they did this back in 2012, which is fantastic. This is 7 years ago. Including restricting the sale of certain kinds of antibiotics in local pharmacies by 2014. But that goalpost has already been lost. So the government committed to this in 2012, set for 2014, and it still hasn't happened. In a 7-year plan, we're 5 years behind. This is not good progress. So it's great for us to set goals and to be having this conversation, but we also have to stick to goalposts because it's not enough to say, well, we said we would do this, we've committed to doing this, we actually have to do it, and we have to do it on time, otherwise there's no pressure for it to actually happen. And at the same time, we're still expecting the use of antibiotics to still be rising dramatically. This is a human problem with a human cost. 700,000 people die every year globally of this issue. And this is a very low-ball issue. 82% of the infections in babies are now showing instances of resistance in India. About 58,000 babies die every year in India of infections that are resistant to broad-spectrum antibiotics. Our babies are dying, our people are dying, and it's going to get worse. By the year 2050, we're expecting 10 million people to be dying of antibiotics every single year. This is the same number of people that die from cancer combined with the number of people that die in car accidents every year. 10 million. This is a crisis. It's a big issue. Before I talked about how antibiotics take us 20 years onto our life expectancy, those 20 years are disappearing. Just like 70 years ago, when people were told, you now have an extra 20 years onto your life, and it dramatically changed everything that it was to live, because you now have 20 more years of life. That's disappearing. It's different for me to come to you and say, yeah, you know how you were expecting to live to be 75? Yeah, now it's actually 55 again. That's dramatically changes how you're going to live your life, and that's the kind of world that we're entering into again. If we do nothing, we are killing these people. So on a lighter note, slightly lighter, we're going to look at things that you can do. Nope. Thank you. How to see the world in 3 easy steps that anyone can do. This was cut off for a reason. I don't want anyone to see the word do. So these are all things that you can do to save the world. Oh, don't get cozy. Well, so step number 1, prevention is better than a cure. This should look familiar, yeah? But you can take part in this too. So if you don't get sick, you don't need to use antibiotics at all. Just like on a global stage, if we're able to reduce how much antibiotics we use, then resistance goes down. So if you don't get sick, you don't need antibiotics. So do what you can to stay healthy. Make sure you're practicing good hygiene yourself and at home. Make sure you're practicing good hygiene, especially especially just like washing your hands. All you need to do, wash your hands 30 seconds with regular soap. You don't need antibiotic soap, just regular soap. 30 seconds. Sing happy birthday to yourself twice as you lather your hands. It's very easy. You look very weird in bathrooms, but it's totally worth it. About 40% of all infections that are transmitted, that are resistant, they're bacteria that really happen in our hands. So if you can cut back on that, then you'll be able to cut back on how much antibiotics you'll actually need to dramatically reduce it. We also know that the agriculture sector contributes a lot to how much antibiotics are being used. So about half of all antibiotics are used in animals instead of in people. Now, the rate of regionally changes on how much antibiotics goes into animals versus going into people. But if you can reduce how much meat you eat, I'm not saying cut it out necessarily if you're not vegetarian already, but if you just reduce how much meat you eat, then that'll dramatically impact how much antibiotics farmers are using, and it'll prevent the rise of antibiotics. So prevention is always the best thing you can do. Next, it works. We can't fix what we don't understand. Now this, again, should look very familiar. If you are not a doctor, don't try to be a doctor. Does that make sense? If you are not a doctor, do not write your own prescriptions. Maybe ask a doctor. If you are sick, talk to a doctor. They know your health very well. They went to school and everything. There's separate universities for them and all of that. There's a reason why they go to university instead of using Google. Don't be your own doctor. Yeah? Are we clear on that? We can't fix what we don't understand. You don't understand human biology like a doctor understands human biology. So don't try to fix it 100% of the time. Taking antibiotics, if you do get prescribed antibiotics as well, make sure that you take the prescription properly. Don't be your own doctor. If your doctor says, take these antibiotics for a full week, and you say, great, and you go home and you start to feel better after 3 days, you are not a doctor. You take it for the full week. That's what your doctor wanted. You want to make your doctor happy and kind. So, do your E. coli some good. Listen to your doctor. And finally, the most important thing about this is that we're all in this together. This is a global issue. It's not an Indian issue. It's not a British issue. It's not an Italian issue. This is a global issue. This is happening all over the world. And it means that we really have to cooperate with one another in order to do it. Bacteria don't look at the world with borders, and we shouldn't either. In the same way, when you look at how you wash your hands for 30 seconds singing happy birthday to yourself twice, and making yourself feel silly, but only a little bit. Also, it's like your birthday every day, which is always nice, to be pleasant. Just like as countries, we need to think of ourselves as being all in this together. We also need to think of being in this together as a people, as human beings. So if you see your parents trying to give you antibiotics without seeing a doctor first, or taking antibiotics without seeing a doctor first, you can yell at them. You have permission from me. You say, no, we're not supposed to do this. See your doctor. You can't fix what you don't understand. You're not a doctor. And we're all in this together. So this is how we should be acting. We need to support each other in order to make this properly happen. Maybe it means that you help your family cut back on how much meat it consumes. There are a whole bunch of different things that you can do to make sure that we're all in this together, that we're all cooperating, that we're all working together. Because that's really, really important. Bacteria function in colonies. They function in communities of millions and billions. And we should think of ourselves as being a colony of millions and billions, all working together and all doing the same things. And that's also why it was so important, why I, as a curator at the Science Museum in London, why it was so important for us to help tell this story in India. because this is a global issue with global stories, global impact. It was so important for us to cooperate with these wonderful partners that we have at the National Council of Science Museums. So I'd also like to thank our partners for being involved in this project and being just so fantastic in helping the project along every step of the way, being so supportive and really doing everything they could for us. So that is everything. I've done boring you. I can take some questions. Like, I haven't forgotten you. The brain.
Sir, if I am able to banish all bacteria from my body, will it affect me negatively?
Oh, so badly. Oh, so negatively. Yes, yes. So we know, for example, that if you have an unhealthy microbiome, that this is still a new area of research that we're still figuring out. And I'm saying that a lot because science has advanced a lot in the last 10 to 15 years. If you were to get rid of the bacteria in your body, that will help you have a lot of trouble digesting your food. I'll say diarrhea. Yeah, you can say diarrhea, right? You'll get a lot of diarrhea. And we also know that an unhealthy microbiome has been connected to conditions like Parkinson's disease, Alzheimer's, food allergies, and in some cases even autistic spectrum disorders. So like there are a lot of ways that we know that the brain and the gut interact a lot. And if the relationship is unhealthy, especially if you don't have a relationship, then that can be really dangerous for your long-term health. Now this is on top of the fact that a lot of bacteria that live inside and on us, they're what we call niche fillers. So if you get, a niche is like a corner, and it fills a little corner. So we have on our skin a bacterium called Staphylococcus Aureus, which is lovely. And it takes up a lot of space on our skin, and it eats a lot of resources on our skin. And it means that if there's a bacterium that might cause disease in us, and it moves onto our skin, it's going to be surrounded by these Staphylococcus aureus. So it's going to have a harder time finding food, finding a place to settle down, and it's just going to be kind of forced out by competition. Now, if we didn't have that Staphylococcus aureus on our skin, that means that a bacterium that wanted to cause an infection and move onto our skin, there would be nothing there to stop it. And so it's really important for us to have bacteria in our bodies. It helps us live healthier lives, and it can prevent future health issues that are more than just infections. Things like Parkinson's.
Well, we're at the end of our program, at the end of our talk. So once again, a big round of applause for this group. Thank you, Mr. PAQUIN.